This course, as you know, will deal with one of the largest group of biomolecules, what we know as enzymes. Now, you are familiar with the number of biomolecules like carbohydrates, lipids, proteins. Now, the enzymes fall into the category of proteins, and the course will cover various aspects of enzymes, particularly in relation to their application as a biocatalyst in the process industries. Now, I think one of the very basic issue which comes to mind is that why do we study enzymes as a separate class, in addition to your study of various biomolecules in other courses like biochemistry. We have a separate course on enzymes, and some of these aspects I will be dealing in today's lecture, which will be basically of introductory nature and also give you the scope of this course. Now, the first issue of the enzymes, that is why do we study enzymes, essentially opens up many features of enzyme molecules, and the first and probably the most important is that what is their role in a living cell, or what is their role in the realm of biotechnology as a whole. If you look at a basic definition or a very broad operational definition of biotechnology, which deals with the application of scientific and engineering principles to the processing of materials by biological agents to provide goods and services. Now, here I like you to mark the term biological agents, and these biological agents cover microbial cells, cellular organelles, enzymes, maybe other parts of the cells like membranes, and a variety of biological material that can be used as a catalyst in process industries. That is what is the determining feature of biotechnology, and among these groups of biocatalysts, enzymes play a very significant role. As a matter of fact, if you go a little deeper into the various activities of a living cell, besides looking at the industrial application, a living cell is a very complex entity, in the sense that it carries out all functions of any chemical process plant that you may be knowing. You can conceive any most complex chemical processing plant, say for example a petrochemical complex, where the petroleum is fractionated into various products, and I like to give you a very simplified analogy of a living cell to that of a chemical process plant. The cell does a variety of functions, that is crushing and grinding, transport of materials, power generation, control of information, transmission of information, and then release of the finished product, assembly lines, release of the finished products into the user clients. All these functions are also basically carried out by a living cell. Now, the key issue is that all these functions in the living cell, may it be a microbial cell, may it be a plant cell, or it may be an animal cell, they are mediated by the catalysts known as enzymes, and therefore, the enzymes play a very central role in the function of living cells, or in other words you can say that they are one of the most important executives in a living cell. Even disturbance in one of the enzyme catalyzed reactions can lead to some abnormalities in the cell function. We are familiar with many of the diseases which are seen when one of the enzymes stops functioning. Similarly, in the case of a living cell, if the enzyme system fails, many of the processes can be disturbed, and ultimately leading to the life cycle of the cell itself. Now, with such an important component, it becomes important to understand how do they perform those reactions, how do they manage the integrity of the whole cell, and the most significant part is that they regulate these functions of the various functions of the cell in such an accurate manner probably no other control system in any chemical process plant can think of. There is a perfect control, fine tuning of the enzyme activities as per the desired requirements, and also we also know that the cells are energetically, in terms of material consumption, are very efficient systems. The wastage is minimum. The energy is generated to the required level, materials are stored in case they are not needed. All those functions are a key part of the living cell, and all these functions are attributed to the function of enzymes, and that is where we get to understand enzymes in a more detailed fashion. Also, we must appreciate that we can carry out the cells for making these functions possible carry out a large variety of reactions. They can range from hydrolysis, polymerization. We are all familiar of how the different components or molecules, intermediates in a living cell are synthesized. They are all they all take place through a series of chemical reactions. Now, these chemical reactions could be hydrolysis, could be polymerization, could be epoxidation, could be reduction, and so on. A whole lot of chemical reactions do take place in a living cell. Now, the same scenario is that even the chemical process industry depends on a whole lot of chemical reactions which are catalyzed either spontaneously or with the use of catalysts. So, the analogy lies that the reactions that are catalyzed by enzymes in the living cells, if they are carried out in an industrial atmosphere with perfect energetically economical situation, environmentally friendly conditions, we have a very good friend in enzymes, and that is what is again another basis of use of enzymes in the process industries, and also the possibility of using them, possibility of studying them in such a great detail. Now, if you look at some of the aspect of enzymes as a process biocatalyst, very broad picture which refers to the role of enzymes as a biocatalyst, we must be aware that all these enzymes function at the ambient conditions, at the conditions at which living cells are known to live. Of course, there might be some exceptions. The organisms that are found under very sort of extreme conditions, they are exceptions, but by and large most of the organisms that live under ambient conditions, they produce or they provide us the enzymes which can function under mild reaction conditions, and that is the basis of their energetic efficiency. That means, the energy requirement of those reactions they catalyze could be very, very meager. They have a very high turnover number, because when we will discuss the reaction mechanism, we will see that they are very efficient catalyst, and when I say very efficient catalyst, our counterpart to compare is the chemical catalysts. There are a large number of reactions which can be catalyzed both by enzymes as well as by chemical catalysts, and the subtle mechanisms that are involved in the enzymatic catalysis make them very efficient catalyst, meaning a high turnover number or a significant reduction in the energy of activation of the reaction. Then a very characteristic feature of enzymes is their specificity. That means, for any given reaction you have a specific enzyme, or the other part vice versa. That means, for any given reaction you need a specific enzyme, and such a specificity essentially is both advantageous as well as disadvantageous. Advantageous in the sense that specificity will bring in lack of any byproducts, because it will not act on any of the other substrate molecule which are present as adjuncts or contaminant in the feed stream, and you will only be reacting the desired substrate converting into product. On the other hand, the disadvantage is from industrial point of view. That means you cannot produce them in very large quantities, because you require small quantities of catalyst for specific reactions. If one catalyst can work on a variety of reactions, the life is simple, as happens in the case of chemical catalyst. For example, vanadium pentoxide or colloidal platinum. Now, they can act as oxidation catalyst for n number of reactions, or ranine nickel for hydrogenation. Well, whatever has to be hydrogenated, it can be used, but the same scenario does not happen. You need a very specific enzyme, and that brings in some sort of disadvantage as far as their role as a industrial catalyst is concerned. The two of the definitely undesirable features of enzyme as catalyst is they are relatively fragile in nature, with very mild deviations from their normal operating conditions, they can be deactivated. That means basically because their whole function is dependent upon a very subtle conformation, and if that conformation is disturbed, and all this conformation is a result of not only covalent interactions, but of a large number of non-covalent interactions which can be disturbed very easily. And that is why under very close reason the deactivation of enzymes can occur, and deactivation essentially from the industrial point of view is some kind of a loss. The other factor is cost, which is again an undesirable feature. Obviously, that is a feature which probably applies to any commodity we want to use for industrial purposes. We will always like to choose a commodity which is less expensive and does the job. And because the enzymes are produced in small concentrations by the living cells, because the only source of enzyme to us are living cells, and they are synthesized in very small quantities. So, obviously, their cost per unit quantity is comparatively very high, and coupled with their stability, it becomes an expensive proposition. But of course, we are also aware that with some of the recent developments in biotechnology, molecular biology and other sciences which involve isolation, purification as well as stabilization, the costs have been brought down, the concentration of the enzyme protein which can be synthesized have gone up, and the situation is changing. Now, another very significant equation as far as the enzymes as process biocatalysts are concerned is between availability and applications. As we saw that they carry out a large number of reactions, but although in a living cell various enzymes are available that carry out these reactions, but their availability in large quantities for commercial applications are limited. And as a matter of fact, only those enzymes which are required in bulk, when I say bulk I am referring to at least if not tons, at least quintals, many of them in tons really, they cannot be produced on mass scale, and their availability for commercial application is limited. There are certain you may be familiar with some of the classical examples of amylases, glucose isomerase, your cellulase, lipase, proteases, which are the enzymes that are produced in bulk and are available in large quantities. Obviously, the other part is there, because of their bulk production, the cost has also come down and become reasonable for commercial applications. Now, the issue of availability is very intrinsically linked with the availability with the applications of enzymes. Unless the applications emerge, one will not like to produce them in large quantities, and of course, the paradox remains, the applications do not emerge till they are available in large quantities. There are a large number of examples which will illustrate my point. Take for example a candidate like lipase, the enzyme that hydrolyzes lipids into the constituents fatty acids and glycerol. Now, obviously, the enzyme is not available to the in the quantities that many of the other enzymes like amylases and proteases are available, but there is a potential it can be produced provided the applications emerge, so that the scale of operation which is required for bulk operation can be easily maintained, and so the reversible interaction between availability and application is a very intrinsic feature, and probably will illustrate the story of many of the enzymes which have become commercially viable today. Now, of course, as far as availability is concerned, there are two major factors. One is that we have to synthesize them microbially. Now, here I would like to again add that, as I mentioned, that theoretically one can produce enzymes from any living source, microbial, plant or animals, but the trend in the recent past has been that the microorganisms have been the preferred choice for a variety of reasons. Some of you may be familiar with them, and the most important has been, one is diversity. That means, and second is that means you can choose a microorganism which will be responsible, which will be able to act on a particular substrate, synthesize the required enzyme. On the other hand, you also have a very clear understanding of the genetic machinery of the cell, a microbial cell, that controls the enzyme synthesis, which you can manipulate, and which leading to hyperproduction of the enzyme. Of course, their bulk production, their bulk growth is very simple. The parameters, the equipment are all standardized, thanks to many other products which are produced microbially. That gives us an edge for microbial biosynthesis of enzymes. The other important part is isolation, purification and stability. And isolation and purification, one of the very characteristic feature is that most of the enzymes are purified by a series of steps to get a electrophoretically homogeneous protein band, which is perfectly pure enzyme. Now, for most of the industrial applications, except for of course some of the medical or diagnostic applications, you may not require a highly purified enzyme. For bulk industrial processing, we have to stop at purity level somewhere in between, compared to a crude enzyme where the specific activity is very low, to a level where the contaminating enzymes which can disturb the process are removed, but not that all the contaminants are removed, which may not be required, because any order of purification will mean additional cost enhancement in the value of the product. As far as applications are concerned, lot of engineering inputs are required, particularly for the extended use of the catalyst. I mean, we do not want to use it just once. As a matter of fact, I think one can take a clue again here from a living cell. The cell maintains a very perfect economy in terms of its energy as well as materials to synthesize the proteins. It does not use the protein only once, it uses in a cycle and over a period of time. Of course, there is always a I mean sort of maintenance part where it has to maintain the protein synthesis, but it is being used repeatedly, and in many of the organs of the cell, the enzymes are supported on certain organelles, so that they behave almost in a fashion of insolubilized enzymes. The similar based on the same concept, one can immobilize the enzyme on a support, insoluble support, what we call as immobilized enzyme, and use them for processes that can be used in continuous mode like any other chemical process. The term very commonly used in the chemical industry, heterogeneous catalysis. Then a very important aspect is bulk environmental design. So far we have been limited in our mind regarding the application of enzymes that they can work only in the aqueous environment, which today is probably not a very perfect statement, because a number of enzymes have been shown to function even in a non-aqueous environment, or in other words more accurately microaqueous environment, where you require a very small concentration of water. Rest of the bulk environment can be replaced by an organic solvent, and that can give us a whole new range of chemical reactions which can be carried out using the enzymes as biocatalyst, particularly a transition from hydrolysis to synthesis, and many of these reactions have become commercially viable today. Then of course, ultimately for any process application of these enzymes, you need to engineer the system. Even after you have done immobilization, even after you have studied the optimum reaction parameters, the whole systems has to be engineered. I mean to say that you have to develop a particular hardware, a reactor, an enzyme reactor in which the whole reaction can be carried out with maximum productivity, because ultimately the whole concept or the whole interest will be on the productivity, and which can be tailored by doing the proper process engineering. And then you may need to monitor certain parameters, the instrumentation, control, then downstream processing of the product, all has to be integrated so as to have a complete application route. In summary, the broad objective of this course, as I mentioned so far, will be to study various scientific, technological and engineering aspects associated with the application of enzymes as a biocatalyst in process industry. Now, this in a very brief terms can be the broad definition of or broad objective of this course. Now, here I think I liked again you to be very critical about the scientific, technological and engineering aspects. A very almost an analogy with the biotechnology in general, even the enzyme technology also has a very multidisciplinary nature. It is not that one discipline, for example, understanding only biochemistry can lead to understanding of the application of enzymes in process industries. A whole range of basic and engineering sciences are required to be understood. Of course, some of them I will illustrate. Now, for example, if you start with, let us say, a basic science like chemistry, many aspects of chemistry have to be understood in relation to enzymes, or in other words, you can add the term biochemistry, the functional aspect of the enzymes, what kind of chemical reaction they catalyze, what is the thermodynamic limits, what is the equilibrium conversions, what are the free energy change, whether the reaction is feasible, the coupling of the reactions so that they become feasible in terms of free energy change, and all those aspects of the enzyme protein will come under the realm of biochemistry. A very important role is played by physics. Today, a large variety of developments that have taken place in the area of biotechnology in general and enzymes in particular can be attributed to the use of a number of physical techniques which have made us to understand a variety of systems and develop or improve upon them for various applications. And physics becomes a very important component to understand the enzyme role. And of course, as a matter of fact, you may be familiar that with the result of these applications and developments, the whole science of biophysics has emerged today. Then you have after chemistry and physics, I think the major scientific component will be biology. I think for any enzyme to be produced in large quantity, you need to identify a biological system which can produce the enzyme. It can be microorganism, it can be a plant, or it can be animal. Of course, as I mentioned earlier, in the bulk of the applications or in the bulk of the cases of enzymes, we look at microbial systems. That is a component which biology will make us understand. We will have to understand its morphology, its physiology, so as to be able to design a appropriate enzyme production system. The role of protein biosynthesis by that organism, which will regulate the synthesis of enzymes, the regulatory mechanisms involved in the biosynthesis of those enzyme proteins again will also be understood through biology. Of course, in the recent years an added dimension is on molecular biology, where we tend to understand the structure function relationships of various biological molecules at a molecular level, how nucleic acids take part in energy transformation, in information transformation from one cell to the next generation. What are the mechanism of cell-cell signaling? They also have an understanding between communication between different cells, or how does the protein molecules behave as a defense molecules. All variety of those functions at a molecular level has become an added dimension, which has been of course at the heart center of the biotechnology in general. So, the similar applications will come here for biology also. Then ultimately you come, I think a very important scientific development has been in the computational science. Most of the industrial applications will require the application of computational sciences to those systems. Today even design of a enzyme protein, modifications for tailoring it to give some desirable properties is a job which can be taken up only with the help of computational sciences. Experimental sciences will take years together to come out with those conclusions which can be brought in by many of the computational programs. Of course, you will need lot of data available to you before you can apply them, but once the data are available, one can easily compute and predict many of the behavior which otherwise would have taken a very long time. That means, for example, site directed mutagenesis. If what happens if an amino acid is removed and another amino acid is inserted in the polymeric chain, in the protein chain, its structural behavior can be very easily understood with a computer graphics, which otherwise doing experimentally probably is a Herculean task. Then these are some of the basic sciences which are important. On the other hand, we have a whole range of engineering sciences which are important as far as enzyme technology is concerned. You will encounter, of course, as I mentioned, you need to design enzyme reactors which can take care of transfer of material, transport of energy, transport of momentum, and all transport processes can be taken care. You need instrumentation for monitoring and control of the reactor. You need a concept of electronics, particularly in some applications like biosensors. Enzyme based biosensors will require transducers which can be, and of course the ultimate is biochips. Of course, we are far from the reality, but still as a concept it has probably attracted the attention of many scientists. That means, can we use the protein molecules or enzyme molecule as biochips in the computers. So, the whole computation becomes based on biological molecules. That is another. So, a large number of engineering operations including mass transfer, heat transfer, material balance, energy balance, product separation, your kinetics, thermodynamics is also involved in the understanding of enzyme science. And therefore, one of the characteristic feature is that the whole enzyme science interfaces with a large number of basic engineering sciences, and it is this interface which makes it so attractive to study, exciting to study, and important to study. It has become today probably for a biotechnologist impossible to leave aside an enzyme molecule, understanding of enzyme molecule in detail before he can call himself to be a professional in the area. Another important aspect which I think I like to touch upon in today's lecture will be on enzyme classification. Now, you will be surprised to know that so far globally about 3000 enzymes have been isolated, characterized and reported in literature. It is not a very large number compared to the feasibility of combinations that are feasible. If you look at enzymes as a polymer consisting of 19 different amino acids and an average length being, let us say, 150 or 200 amino acids linked together, 19 different amino acids, probably the possibility of variety of diversity of the proteins that are feasible is enormous, almost approaching to infinite. As against that, we have been able to isolate and characterize only about 3000 of them. Now, this 3000 number, just to understand and bring it into a systematic feature, have been classified into 6 different classes. In fact, this job was done way back in early 70s by International Union of Pure and Applied Chemistry, what we know as IUPAC. They gave a classification system which is acceptable globally today, and now it classifies all the enzymes into 6 major classes: oxidoreductases, transferases, hydrolases, lyases, isomerases and ligases. Now, a whole range of chemical reactions or biochemical reactions that take place in living cells are mediated by these different classes. Of course, you must know that some of the reactions are not independent, they are coupled and thereby producing an effect which could be, for example, say a polymerization reaction. Now, directly the polymerization does not take place as it is mediated by series of transferases, and then ultimately the transferases and ligases, then ultimately you end up with a polymer of different monomers. And so, but the basic reactions, basic enzymes can be classified, and each of these class are also characterized in subclasses. Subclasses refer to the type of substrate or the type of product that is produced by those enzymes. So, basically the whole range, I have given some of the examples of each of the class here. Oxidoreductases, a bulk of the oxidases, reductases and dehydrogenases fall under this category. The transferases involve group transfer reactions, where one of the functional group, let us say a substrate molecule is phosphorylated by reaction, usually they are Y substrate reactions, involve 2 substrates. Hydrolases and by and large the most commonly employed industrial catalysts, mainly because of the simplicity, because the co-substrate is water which is present in large quantity and one can use them for hydrolytic reactions. Now, by changing the bulk environment, the hydrolases can be tailored to be used for synthetic reactions as well. Then lyases, again some kind of an additive enzymes in which which catalyze the reaction elimination reactions that result in the double bond formation. You have isomerases, another important class of industrial enzymes which isomerizes one molecule into the other isomer of the molecule. One of the most significant enzymes under this class is isomerization of glucose to fructose. Ligases, of course, join 2 molecules at the expense of energy source. Of course, as you know, in most biochemical reactions, ATP is the energy currency which is consumed and of course regenerated through catabolic processes, and such reactions are classified under ligases. In fact, this is a classification, as I mentioned, based on the IUPAC nomenclature, more from the scientific point of view, but from the industrial point of view or from the application point of view, another important type of classification which is made is in terms of, they are in terms of 3 classes, what we know as bulk, diagnostic and therapeutic. Of course, this classification is essentially based on the cost of the enzymes. In fact, cost of the enzyme, and as you see in this figure, the cost is directly linked with the concentration in the starting material. It could be a fermentation broth in most cases. Now, in the fermentation broth, if the concentration of the product or the desired enzyme is very, very low, the cost is very high. And of course, the second factor of cost is the application, the degree of purity you need for that particular application. Now, you know, for the bulk enzymes, as you can see here, they fall in this category. The concentration in the starting material is of the order of about 1 kg per meter cube, which is significant enough to recover, and you do not require for in most cases a very high degree of purity, and therefore, their cost is also reasonably low, say for example about $1000 per kg. The next category is diagnostic. It includes also the analytical enzymes. Here the concentration is of the milligram per liter, grams per meter cube level, much lower. Degree of purification required is also larger, because you will not like any contaminant to be detected by the protein contaminants present in the sample, and their cost is therefore significantly high, of the order of about 10 to the power 6 US$ per kg. Of course, I must like that you should not get threatened by the amount of cost 10 to the power $6. The quantities required are also in micrograms, they are not in kilograms like other molecules. So, that is where they are affordable. The third and probably the most expensive class is therapeutic enzymes, which are exclusively required for medical application, almost like life saving drugs, where cost is immaterial. You do not make choice on the basis of cost. In the case of industrial operations, we always make a choice between two alternatives on the basis of cost and of course effectiveness, but if the effectiveness is same, the cost is a principal determining factor. Whereas in the case of therapeutic products, the choice is not on the basis of cost, but on the basis of its need. If it is a life saving drug, many of the therapeutic enzymes, particularly the recombinant proteins that are available, some of the enzymes like streptokinase, urokinase, they are blood clot dissolving enzymes, which can within fraction of a minute can simplify the situation of a say a cardiac failure problem due to blood clot, which we can immediately act and they can be solved. Similarly, the number of other such therapeutic proteins or enzymes are available which function in those, like asparaginase for patients suffering from leukemia is another life saving, and similarly their cost is also of a very high order. Of course, the quantities again required are of less than microgram quantities, but in a very highly purified state. So, in fact, this is what I had the last phase of this today's lecture will cover on course outline and references, and the course will broadly cover these aspects. I have given you some idea about the introductory remarks on the enzymes as a process biocatalyst. We will go into little more details on the chemical nature of enzymes, that means enzymes as proteins. We will go into functional nature of enzymes, enzymes as a catalyst, what makes them such a efficient catalyst, what are the factors that are responsible for their deactivation, what are the approaches that are available for stabilizing the enzymes, what is the role of active sites or ligand binding site on the enzymes. Those issues will be dealt in functional nature. Then we will go on the kinetics of enzyme catalyzed reactions, because the reaction kinetics will play a very important role in the choice of enzyme reactors, to understand the reaction rates and the role of associated physicochemical interactions that will take place once you immobilize the enzyme or use them in a bulk environment other than water. Then I think one of the most significant tool which has made many of the enzyme reactions commercially viable is immobilization of enzymes. A large number of techniques are used, we will discuss them in detail. The use of enzyme reactors, the types of reactors that are possible to be used with immobilized enzymes, with the soluble enzymes, their characteristic features, comparison between their performances, how to choose the enzyme reactors, then mass transfer in immobilized enzyme reactors, because immobilization of enzyme will have some additional physicochemical features like diffusion, partitioning. Now, those issues when they are placed in a reactor, how do they influence the performance of the system. Immobilized cells, very often in the if the enzymes are intracellular, it has been felt that for many applications their isolation and purification is not required. You can immobilize the whole cell as long as the cell does not have, or you can inactivate other contaminating enzymes in the cell which will interfere in the process. Then ultimately in the case of bioprocess design, some of the basic design principles for the enzyme catalyzed processes, a few case studies I like to illustrate some of the earlier issues with respect to specific cases, and ultimately I think I like to take up enzyme based sensors. The enzyme sensors have become an important activity where immobilized enzymes are used, both immobilized enzyme and immobilized cells are used, and they have become very important tool both in diagnostics as well as monitoring of information from reactions. So, this will be our ultimate course outline.